to model in vivo tissue compliance using acrylamide hydrogels. This is accomplished by first generating reactive bottom cover slips and siliconized top cover slips. The second step of the procedure is to pour and create the acrylamide hydrogels. The third step of the procedure is cross-linking the extracellular matrix protein of choice to the hydrogel. The final step of the procedure is incubation and analysis of cells. Ultimately, results can be obtained that show how varying extracellular matrix compliance in vitro regulates cell behavior through analysis using immunofluorescence microscopy, real-time quantitative PCR, and Western blotting.
Today we will show you the procedure for the generation and use of acrylamide hydrogels.
We use this procedure in our laboratory to study how extracellular matrix stiffness regulates cell morphology, cell signaling, and proliferation. So let's get started.
Begin this procedure by generating reactive cover slips. First, place a layer of parafilm on the bottom half of a 150 mm Petri dish. Then, transfer up to 9 autoclave 25 mm cover slips onto the parafilm and cover them with 1 ml of 0.1 molar sodium hydroxide. Incubate the cover slips for 3 minutes. Following incubation, aspirate the sodium hydroxide with a vacuum line. Working in a chemical hood, pipette 0.5 ml of 3-aminopropyl trimethoxysilane, or 3-APTMS, on each cover slip. Incubate the cover slips for 3 minutes and then aspirate the 3-APTMS.